If you're tuning in for the first time, welcome to the Serious Talk. It is the quarterly webinar series hosted by Claret that showcases the impactful work of our customers and collaborators and friends, often using our flagship NGS Prep seriously. And today it is my pleasure to welcome Dr. Sethu Pitchiaya, who is an assistant professor in the Departments of Urology and Pathology at the University of Michigan. Dr. Pitchiaya's research sits at the intersection of molecular biology, cell biology, chemical biology, biophysics, where his lab combines cutting edge technologies, including single molecular microscopy, super resolution imaging, and so on, to understand how cells respond to stress in both health and disease. His work explores how changes in subcellular organization and gene regulation influence cell fate decision, which is, of course, important in cancer and other diseases. His research has been recognized with numerous awards, including the NIH Outstanding Investigator Award. And he has published in leading journals, including Nature Genetics, Molecular Cell, PNAS, and so on. Today, he'll share how his team is using integrated imaging and multiomics to uncover how cells sense and respond to stress and how these mechanisms lead to new insights into cancer and other disease. Before we begin the presentation, there are some house rules. Please feel free to post your questions in the Q&A section that you find at the bottom of your screen during the talk. We'll read out the question at the end of the talk, and Dr. Pitchiaya will answer the questions then. This webinar is recorded, and all attendees will receive the recording in about a week. We will also include a discount code because an attendee discount code that you could use in your next Seriously purchase. With that, I'd love to hand this over to Dr. Sethu Pitchiaya. Welcome again.
Oh, thank you so much for this kind introduction and for the opportunity to present some of our work. So today I'm going to be talking to you about an integrative interrogation of cellular stress response in both physiology and disease. So the lab is largely interested in understanding one fundamental question, that is, how do cells deal with stress? And when I say cells, the work that we do is relevant to many cellular and tissue contexts. We are currently interested in cells and tissues pertaining to the genitourinary lineage as well as the neuronal lineage. And when I say stress, I'm being fairly agnostic here. The lab is interested in understanding the response of cells which face a variety of perturbations which pushes them away from the state of homeostasis. And when such perturbations occur, cells make one of three fundamental decisions. That is, they give up or die, they can survive, or they can thrive. And depending upon the decision that cells make, it means very different things in both the physiological and pathological scenarios. For instance, if cells are frequently dying in response to stress, that's a hallmark of several degenerative diseases. On the other hand, if cells can thrive in a harsh microenvironment, that's the hallmark of cancer. So we want to understand how cells within complex tissues make these foundationally distinct decisions. So we look at cellular stress response and decision making in two distinct lenses. One of the lens is based on advice that you or I are given when we are stressed, which is, hey, you know what? Take a pause, maybe do some yoga or focus on things that you really need to get done. And our cells do very similar things in the sense normally they express lineage genes, which are genes that keep a heart cell, a kidney cell, and housekeeping genes that drive cellular function. But when they are stressed, they actually take a pause on all of those and focus on expressing genes that are relevant to counteracting the stress at hand. And what we want to understand is how cells achieve this transcriptional and translational selectivity during stress and how this contributes to cellular decision making during stress. The second lens that the lab takes in looking at cellular stress response is also based on advice that you or I are given, which is, you know what, great, you've taken a pause. Now maybe go hang out with other people, or get a little dynamic, hit the dance floor again, whatever helps you, right? And our cells do very similar things in the sense we now know that they accumulate a significant fraction of their proteome and transcriptome at the very least into what can be described as membrane-less compartments. And there are a variety of them which form in distinct cellular compartments. And these membrane-less compartments, the persistence of these can be explained by a physicochemical principle called liquid-liquid phase separation. It's known that a variety of biomolecules form these membraneless compartments, also called as condensates. And it's known it's a feature of stress response. For instance, I can name one protein that actually forms these condensates all the way from yeast to humans to plants. What is not known is their function. So our lab seeks to understand how these condensates contribute to cellular stress response and decision making during stress. With that, I'm going to dive into two distinct stories. The first one is trying to understand the response of prostatic cells to castration. And this is a highly collaborative work. It was led by two fearless bioinformaticians, Hannah and Yuping, and it was done in collaboration with Dr. Evan Keller and Arul Chinayan. So prostate cancer is one of the leading causes of death in men. 1 in 8 men will be diagnosed with the disease in their lifetime. It's now known that the number of new cases every year is close to 300,000, and the number of deaths is almost 10%. And that statistic is even larger worldwide. So there is an urgent need, especially considering that many men succumb to the disease, to find ways of countering this. And if you look at the disease progression, typically it's diagnosed via biomarkers like PSA. And when your PSA gets elevated, you receive some form of primary therapy in the form of surgery or radiation. Many patients actually benefit from it. However, several patients actually end up getting the disease back, at which point androgen deprivation therapy, countering or targeting the androgen receptor, a key driver of prostate cancer, is one of the frontline therapeutics. And again, androgen deprivation therapy has been super useful and has been instrumental in reducing the disease burden. However, a significant fraction of patients develop a resistant state and progress to a state of the disease called castration-resistant prostate cancer, or CRPC, which is almost always lethal. So there is an urgent need to understand how castration resistance occurs, and if there are markers that we can use as predictive, with predictive values to predict which patients will develop to this form of the disease, and also find therapeutics that counteract this disease. Uh, there are several mechanisms that have been posited, both genetic and epigenetic, and we were primarily interested in the epigenetic mechanisms by which castration resistance is actually driven. To this end, we resorted to a classical model of studying castration, which is castration response, which is the mouse model of castration, where the mouse prostate is intricately organized into these distinct lobes, with the anterior prostate, the dorsal prostate, the lateral prostate, and the ventral prostate. And if this prostate is receiving very less androgens, testosterone especially, via castration, the prostate involutes, which means several cells die and very few remain alive. So this gives us a state at which we can actually look at how those cells that are surviving are actually responding to castration, and whether these have any bearing to castration-resistant prostate cancer. So to do that, we wanted to look at every individual cell type in the mouse prostate, and again, the mouse prostate, and which has which are organized into these individual glands in every single lobe, is very similar. The glands are very similar in humans as well, and each of these glands have several distinct cell types, and they have at least four distinct epithelial cell types and four distinct stromal cell types, so eight cell types in total. Uh, Isolating them individually and studying them is a great way of doing it. We resorted to for a more discovery-based approach. So sequencing is one of the best ways to do it. However, bulk RNA-seq is a way is super useful. However, it's very similar to how you actually consume a smoothie, where you take a whole bunch of fruits, just crush them together, and then you have a nice smoothie with different ingredients in it. You just can't identify the individual ingredients in it. So in this particular case, it's very difficult to identify the individual cell types and see how each of them or each of their gene expression changes in response to castration. And there is certainly a solution now, which is single cell RNA sequencing, which is very similar to having a fruit bowl, where you could easily identify what are the individual fruits in the bowl and know what you're consuming. And there is also much more sophisticated technologies of spatial transcriptomics, where not only do you know what the ingredients are, it's very similar to a fruit pie, where the fruits are placed in distinct locations in the pie, and you really know where each of the fruit is as well. So we combined single cell sequencing and spatial omics to study castration response in the mouse prostate. So first, we use single cell RNA sequencing. And the way it works, you take the tissue, the prostate, you dissociate it into individual cells, you barcode them so we know what each individual cell is, which can be sequenced, and then we can get the transcriptome from each individual cell. And what you're seeing on the right here is a UMAP plot, which is essentially a reduction, a dimensionality reduction of all the transcriptomes of each of the individual cells, where each dot is a single cell that you're seeing, and groups of dots which cluster together. For instance, here number 14 or Fib1 or fibroblast, they cluster together, they have very similar transcriptomes. And if there are groups of cells that are far away from each other, for instance, the fibroblast here with LE1, they have very distinct transcriptomes. So they are very distinct cell types. So now, based on this visual cue on a UMAP plot, what you may be able to appreciate is that there are over 22 distinct cell types in the mouse prostate, as opposed to the 8 that was known. So the mouse prostate is actually super complex. So we are glad that we did not use just bulk RNA-seq at this point. The single cell RNA-seq has opened up a new avenue to new cell types that we can start studying and how they respond to castration. We then subjected these prostates to single cell multiomics, which is a combination of ATAC-seq as well as nuclear RNA-seq from the same nucleus. So what you will be able to get from this, as opposed to single cell RNA-seq, which tells you cell phenotyping, is here you can get gene regulatory information. I'm just showing you an example of a track here, which corresponds to a genomic region of PSCA, prostate stem cell antigen, a stemness-associated molecule. Each row is a cell type, where BE stands for basal epithelia, and LEs are the different luminal epithelia, with the numbers denoting the distinct cell types. And in each row is a track corresponding to the accessibility or ATAC signal. And as you can see, in this particular genomic region, the accessibility is highest for LE5. And what you may also be able to appreciate, since we can measure RNA from the same cell, is that it's in this cell that you're actually able to see the expression as represented by this violin plot of PSCA. And what additional aspects we can do from this is we can actually go and look at sequence motifs at these distinct open chromatin and identify which transcription factors may possibly be playing a role in the expression of this gene. And when we do so, we actually identify a motif for KLF5, again, a stemness associated transcription factor, which has a high accessibility, which could potentially be driving the expression of PSCA. So this is great. This is one genomic locus, but now we can actually get this for the entire transcriptome. And what you're seeing down here is a heat map bubble plot where each row is a transcription module, which is essentially a transcription factor and all the genes it regulates. And each column is a distinct cell type. And if you have a large bubble with a red background, so a large bubble means high accessibility for that particular motif, and a red background means high expression of the genes that that transcription factor or module regulates. So you can see, for example, in basal epithelium, TRP63 has a pretty high accessibility as well as its transcription module being overrepresented, suggesting that this is potentially what drives, this transcription factor is what potentially drives the specification of basal epithelia in the prostate. And what came to us as a surprise was when we actually look at those luminal epithelial cells, each of them have a very distinct transcription module that specifies those cell types, suggesting not all luminal epithelial cells in prostate glands are the same. So now that we have regulatory information and we have phenotype cells, we wanted to look at it in the spatial perspective. To this end, we applied spatial transcriptomics based on 10x Visium platform. So this is essentially a proprietary slide that has capture oligos onto which you can place your tissue of interest and transfer the RNA from your tissue of interest to the capture oligos. And the capture oligos are barcoded in space within individual spots, so you know which spot has which particular RNA. And now what you can do is you can get that RNA out. You can do a library prep and actually sequence them, and now you know where exactly in space the RNAs came from. This is not a single cell spatial transcriptomics platform because your tissue can just land on any random spot, and in one spot it's about 55 microns in size. So it's highly possible, depending on your cell type, that multiple cells are there, or it's also possible if your cell type is large that multiple spots are composed of a single cell. So what it does give, though, is spatial profiles across large tissues. In this particular case, the mouse prostate fits within this approximately 0.5 centimeter by 0.5 centimeter area on which this tissue sits. So you can actually get profiles of transcriptome across the entire tissue. And in this particular case, with the mouse prostate, it is actually very easy for us to identify what the individual lobes are just based on the histology sections where distinct lobes, as in the ventral prostate or VP, the lateral prostate or LP, the anterior prostate or AP, and the dorsal prostate as DP, all have distinct histological or morphological assessment. So the pathologist can easily demarcate what those individual lobes are, and that's what you're seeing on the top. So based on that, after spatially annotating regions of the prostate, we use cues from our single cell RNA sequencing and single cell multiomics and applied gene sets as expression markers for distinct cell types and wanted to see how they are spatially oriented. And what came out to us was super surprising. For instance, LE1 or luminal epithelial 1 is largely restricted to the anterior prostate. LE2 or luminal epithelial 2 is actually present in both the dorsal as well as the anterior prostate. Luminal epithelial 7 is largely restricted to the ventral prostate. LE8 is largely restricted to the lateral prostate, and LE5, which if you remember was the cell type which was expressing prostate stem cell antigen, which was KLF5 driven. So this is the stem-like cell type. It's largely overrepresented in the proximal urethra, as well as very rarely in several of the actual prostate lobes, with the highest expression being in the ventral prostate. If you were to look at some other cell types like fibroblasts, they are expressed in almost all the lobes and in fact even in non-prostatic regions, suggesting it's actually these epithelial cell types that have dramatic regiospecificity to anatomical locations, suggesting that this is probably developmentally driven. So now that we had identified the cell types or the ingredient list of cells in the mouse prostate, what are the genes that are actually regulating each of these cell types and specifying them and where they are located, we now ask the question of how do these distinct cell types respond to castration. And when we do so, here is again a UMAP plot, and what you're seeing is a combination of the data from both from prostates that were extracted from mice that were either sham operated or castrated. The sham operated are ones that are called intact, and the castrated are ones that are called cast. And this is a combined UMAP plot where on the left plot, the cell types are colored are only in the intact sample. Whereas on the right plot, the cell types that are colored are only in the castrated sample, but they are grouped together. So what you may be able to see is if cell types from both the intact and castrated samples are close to each other, they're very similar. If they are far away from each other, they're very different. For instance, these fibroblasts, if you see in both the intact as well as the castrated, they're pretty close to each other. So their transcriptomes are very similar. On the other hand, if you look at this luminal epithelial cell type 1 and 2, they sit right at the bottom here in the intact samples, whereas they move dramatically away from their original position in the castrated samples, suggesting that their transcriptomes are very different. So the prostate epithelial cell types in the prostate undergo dramatic reorganization transcriptionally upon castration. Now we can start using our multi-integrated multi-omics approach and use single-cell multi-omics to look at, again, transcription factors that are essentially driving the expression of specific genes in response to castration. So when we do so, this is just an example of a transcription factor called FOSL2 with in luminal epithelial cell type 7 or LE7, which is the ventral prostate associated luminal epithelial cells. And what you're seeing here is an enrichment plot, a meta plot, where we actually look at the accessibility signal or attack signal across a particular motif, in this particular case, FOSL2 motif, which is located here in the 0 position in a plus or minus 200 base pair window. And what this gives us is the footprint of the transcription factor. And as you can see, the footprint of the transcription factor that FOSL2 leaves is higher upon castration, meaning it's gained. And it's relatively lower in the intact sample. So this can also be represented in space. And what you're seeing here in the spatial transcriptomics plot is that this FOSL2 transcription module, as in the transcription factor and the genes it regulates, are overrepresented across the entire prostate. As a sanity check, we can actually look at the androgen receptor, which is what is directly being targeted by castration because you're reducing testosterone, which is what actually binds to androgen receptors. So if you look at it in the same cell type, LE7, in the intact samples, the footprint of the androgen receptor is much higher than that in the castrated sample. And when you look at this in space, again, the expression of the androgen response transcription module is way higher in the intact sample and way lower in the castrated sample. So this is a great sanity check. So now if you look at these transcription modules across various cell types, as well as looking at various different kinds of transcription modules, a pattern pops up. What we see is, in addition to stress-responsive transcription factors and modules being overrepresented, stemness-associated transcription modules, as exemplified by KLF6, also go up across the entire prostate upon castration. Whereas lineage-specifying transcription for modules such as BHLHA15, those are transcription modules that we think is a specification transcription factor for the ventral prostate, actually goes dramatically down. So this suggests that stress-responsive and stemness-associated transcription modules are actually induced upon castration, whereas lineage-driving transcription modules are actually repressed. And this is not just to these two specific transcription module. If we actually look at transcription module that we annotate as induced or higher in the intact sample, are higher in the castrated sample. So the ones in green at the top are higher in the intact. The ones in yellow are ones that are higher in the castrated. They actually have almost one-to-one correspondence in the human data sets where we're actually looking at prostate cancer patients who have either not received any therapy or have received androgen deprivation therapy, where if you see the ones that are in the intact column are higher than the ADT, and these are the ones that we annotated as being higher in the intact prostate, whereas the ones that are higher in the ADT samples in human patients are the ones that are also higher, or we annotated as castration responsive elements in the mouse. And specifically, I'm highlighting FOSL1 and KLF6 as stress responsive and a stemness associated transcription module that is induced during androgen deprivation therapy. We can actually look at this in spatial transcriptomics data from human prostate cancer patients, where when you're comparing treatment naive to androgen deprivation therapy treated patients, both the KLF6 as well as the FOSL2 transcription modules, that is the stemness as well as the stress responsive transcription modules, are highly induced in the androgen deprivation therapy treated patients. And this is not just during androgen deprivation therapy. A subset of these transcription modules are in fact specifically elevated in castration-resistant prostate cancer. Again, I'm highlighting FOSL1 and KLF6 here as representative ones, but there are many others. And these are not elevated in a different type of a prostate cancer called neuroendocrine prostate cancer. So what we have done is specifically identified markers of androgen deprivation therapy response, as well as potential markers that may be driving castration-resistant prostate cancer. So in a nutshell, what we have done is we've actually used cellular cartography, just like how cartographers go across the globe identifying and making maps. We've actually made a map of the mouse prostate. And what we have learned is that prostatic cells typically are driven by specific lineage driving transcription factors normally, but upon castration, they actually stop doing all of those and actually focus on expressing genes or transcription factors, gene regulated by transcription factors that are responsible for stress response and stemness. And this is not just associated with the mouse, but it's actually present even in prostate cancer patients. So where are we going next? We are trying to understand the mechanism by which this happens. Do these transcription factors and gene sets that we have identified have any diagnostic potential? And is there a way for us to target these distinct transcription factors as well as genes that we've identified as a therapeutic for castration-resistant prostate cancer? So having said this, I'm going to switch gears and go to a second story, which will be a lot more mechanistic and molecular in detail, where we are looking at how condensates probably tune transcriptional stress responses and how this could be stress type specific. And this is a study that was led by three wonderful individuals in the lab, Jeff Dudley, and an MSTP medical scientist training program graduate student who just recently graduated, Joel Behrens, who's a clinical research fellow in the lab, and Chayande, a postdoctoral fellow in the lab. So protein homeostasis is central to cell survival. And when I say protein homeostasis, the protein quality and quantity has to be maintained. Protein quality is maintained by other proteins called chaperones that help refold unfolded proteins. And protein quantity is controlled by molecules like the molecular complexes, like the proteasome, which actually degrade proteins. And this is essential for cellular homeostasis. However, when cells are hit with stress, this basis is tipped towards the direction of unfolding and perhaps even the formation of aggregates. And it's well known that these aggregates are largely associated with cell death, much like how aggregates are found in neurodegenerative diseases. However, cells have evolved an evolutionarily conserved mechanism where in response to stress, you have an induction of chaperones and the proteasome, which tips the balance back to homeostasis. And this is driven by several components, including one critical one, which is a transcription factor called heat shock factor, which gets activated upon stress, binds to DNA elements and drives the expression of genes like the chaperone, which feeds into this homeostatic pathway. So overall, HSF function is critical to cell survival. Uh, it's well known that reduced HSF function is largely associated with reduced lifespan as well as several degenerative diseases. And I'm just showing you here a couple of examples. One on the left is a study done in C. elegans where just the expression of HSF1 can actually control the lifespan of worms, where higher HSF1 leads to higher lifespan and lower HSF1 leads to reduced lifespan. On the other hand, on the right, I'm showing you here a Western blot of neuronal cells of brain tissues from patients who did not have any disease or control or had some form of a Lewy body disease. And there you can see that there is reduced HSF1 expression, and this is correlated with the enhancement of cleaved caspase-3, a marker of apoptosis. So overall, dampened HSF1 levels are associated with shortened lifespan and neurodegenerative diseases. On the other hand, elevated HSF1 levels as well as activity is a hallmark of cancer. For instance, I'm showing you here an example of patient survival in osteosarcoma all the way to the left, where higher the HSF1 expression, lower is the patient survival. And in the middle, I'm showing you here survival from breast cancer patients, where higher the signature of HSF1, as in the genes it's actually regulating, lower is the survival of breast cancer patients. And in the recent study that I just showed you in castration response and castration-resistant prostate cancer, heat shock factors are elevated during androgen deprivation therapy, and in fact are elevated during castration-resistant prostate cancer as well. And this kind of makes sense because cancer cells have to constantly divide, and on an average, they make at least 10 to 1,000-fold more proteins per unit time as a normal cell. So what they need is to be at an elevated state of protein homeostasis. So they have a huge protein burden, so they need more chaperone, more proteasome, and this is driven by HSF1 driving the expression of those genes. So this is great. So HSF1 is critical for homeostasis and has huge bearings in both degenerative diseases as well as in cancer. But if you look at HSF1 histologically or pathologically or inside cells, they actually end up accumulating in these foci, which we now know as condensates in the nucleus of several types of cells. It's conserved all the way from yeast to humans. And this process of condensation happens during stresses like heat shock. And here is one example on the top left from yeast. On the top right, I'm showing you some examples of several different cell types subjected to human cell types subjected to heat shock, and you can see HSF1 accumulates within such foci in these cells. It's not just heat shock. It's known that if you subject human cells to ACA or azidothine carboxylic acid, a plant metabolite, but a human toxin, HSF1 accumulates, which is a proline analog, so it can cause protein misfolding. You can see HSF1 forms these nuclear condensates. It's also shown that upon poisoning with heavy metals such as cadmium, you can see HSF1 accumulating in these foci. It's not just that. If you hit cells with proteotoxic drugs like inhibiting the proteasome, again, you're leading to proteotoxic buildup or unfolded protein buildup, you actually see HSF1 in these foci. And finally, it's been shown that in patient tumors, HSF1 forms these foci in the nucleus only in tumor cells, but not in the stromal cells, suggesting there is some form of specificity associated with these condensates and their potential function. So what we asked was what these condensates are doing. And as a biophysical chemist, having things in close proximity within something like a condensate can have two divergent outcomes. One is you bring reactants together to induce chemistry in a way products are made way better, or you actually sequester one of the reactants away from the other in a manner that chemistry goes down. So for a transcription factor like heat shock factor, it's possible that HSF1 gets accumulated within these tight compartments where it's driving the expression of genes, or it could be that HSF1 gets sequestered in compartments where you actually don't get any expression of those genes. So we started to test this out using super resolution microscopy. And what you're going to see here are all HSF1 as well as several components of the transcription machinery, all in their endogenous state. We use immunofluorescence to actually visualize them at nanoscale resolution. For instance, here's an example of looking at HSF1 and RNA polymerase II. And what you're seeing here is that both these molecules are relatively diffuse in the nucleus of this particular cell. However, if you hit them with stress, you actually see them co-condense with each other. So every other molecule that I'm going to show or marker that I'm going to show you from now on are relatively diffuse to begin with, but are probably going to come to a condensate later. So if you look at RNA polymerase II, that's phosphorylated in this fifth position of the C-terminal domain, RNA polymerase 5P, it's an indicator of RNA polymerase being primed for transcription. That co-enriches at these condensates. If we then thought, okay, this is great. Actively transcribing RNA polymerase II is there at these condensates, and HSF1 is a transcription factor that possibly binds promoter regions, which are marked by H3K4 trimethylation. So we thought, okay, maybe these are condensates of promoters, but when we look at H3K4 trimethylation, it is actually not enriched at condensates. So this led us to posit that it's possible that we are looking at enhancers here, which are marked by potentially H3K27 acetylation. And when we look at this in conjunction with HSF1, we actually see that H3K27 acetylation co-condenses or is present at these HSF1 condensates. If we look at BRD4, a bromodomain molecule that actually binds acylated histones, that is also enriched at HSF1 condensates. If you look at mediator complexes, which you can think of as being a staple between the promoter and enhancer, which binds transcription factor and BRD4 together, also co-enriches at HSF1 condensates. And in fact, we're looking at nascent RNA, and here we are looking at RNA of whose sequence we do not know, but these are all metabolically labeled. So we actually can see what is being newly made, and you can see that all of them are actually co-enriched at HSF1 condensates. And this is not just we are looking at them, we actually calculate what is called as an enrichment index. And what you will be able to see is that a lot of the transcription apparatus are actually including RNA are co-enriched at HSF1 condensates, whereas markers of heterochromatin and just histone alone are not, suggesting that HSF1 forms these transcription hubs during acute proteotoxic stress. So we then asked the question of, is HSF1 required for these hubs? So we created a cell line that lacks HSF1. And now without looking at HSF1, we looked at all the other transcription apparatus. So here I'm just showing you an example of BRD4, which forms these nice punctate structures upon heat shock in these cells, parental cells. But if you remove HSF1, it actually becomes completely diffuse. And in fact, it's not just BRD4. If you're looking at H3K27 acetylation, MET12, or RNA polymerase II, you actually don't see them in the condensed state in the knockout cells, suggesting that what was originally an enhancer hub is not formed anymore or is dismantled when you actually remove HSF1. So what does this mean for the cell? The cell cannot make these chaperones. As you can see in the knockout, the expression of HSP-A1A or HSP70 is dramatically reduced. And in fact, the cells themselves cannot survive a proteotoxic stress like heat shock, where the knockout cells have a much larger cell death as compared to the wild type parental cells. So proteotoxic stress-induced transcription hubs require HSF1. We then asked, okay, this is great, but how do these condensates form? So to do this, I'm just going to quickly introduce you to a concept that we borrowed from the omics field, where we create these meta images. What this means is we actually take cells, we look at condensates in different regions using computer vision, we've identified them, and then we just place them on top of each other or make an average image from all those condensates. So what you will get is an average image from these condensate regions, like a meta image, which you can actually see as being, which will help us give a visual cue of something that is being enriched or not. So using this meta-image approach, for instance, under normal conditions, you can actually see HSF1 in green, co-enriching with MET12 in red, and it's the same for HTK27 acetylation, BRD4, and RNA polymerase 2. However, if you inhibit H3K27 acetylation with A485, a P300 inhibitor, you can see HTK27 acetylation goes down, because of which BRD4, which binds HTK27 acetylation, also goes down. RNA polymerase 2, which potentially gets recruited by BRD4, also goes down. So what this tells us is in either of these cases, HSF1 condensates are actually not hampered at all. And this is the same when we actually do BRD4 inhibition as well, where you can actually see that BRD4 inhibition only reduces BRD4 and RNA polymerase 2, but does not affect the previous step of HTK27 acetylation or even MET12 co-enrichment. And HSF1 condensates are not affected here either. So this suggests that it's possible that there is a stepwise condensate organization where HSF1 condensates form, and then they start recruiting MET12, HTK27 acetylation markers via P300 enrichment, as well as then comes all the transcription apparatus that makes this entire compartment transcriptionally active. So this is great. This happens during heat shock. Is this the same in response to all the other stresses that we tested? And what we found was something very peculiar, where in certain stresses like heat shock or chaperone or proteasome inhibition, you have these transcription hubs where HSF1 co-enriches with RNA polymerase 2 and nascent RNA, whereas in several other stresses, such as arsenic poisoning, arsenic trioxide, which is a chemotherapeutic, chemical hypoxia, cadmium poisoning, oxidative stress via hydrogen peroxide, methyl glyoxide stress, which is a metabolite that is frequently upregulated in diabetes, and acidine carboxylic acid, which is a plant toxin, a plant metabolite, proline analog, which is toxic to humans. You actually see that HSF condensates form, but they are not transcriptionally active at all. And to tell a long story short, what you're seeing here are those meta images from all these conditions, where each row is a marker. For instance, this is a phosphorylated 320, a marker of phosphorylation of HSF1, HTK27 acetylation, BRD4, and so on. And each column is a stress. So if you look at it, upon heat shock, you actually gain these phosphorylation marks as a marker of activation, whereas the amount of activation marks for HSF1 is slightly reduced in response to all these other stresses. Again, during heat shock, you see the formation of this enhancer hub, which has all these transcription apparators, whereas in stresses like ACA, you actually see that it has almost everything in place up until RNA polymerase 2 5', but it does not have actively elongating RNA polymerase 2. If you look at a stress like cadmium chloride or cadmium poisoning, you actually see that it has almost everything, but does not have RNA polymerase 2. If you look at stresses like chemical hypoxia or oxidative stress, they actually only have the mediator complex. And if you look at other stresses like methyl glyoxide or arsenic trioxide, the chemotherapeutic, they actually, again, the condensates are there, HSF1 condensates are there, they actually don't have any of the transcription apparatus. This suggests that distinct stresses actually are stalled at distinct stages of this hub assembly. And this is what actually gives to potentially distinct phenotypes. And we can actually test this. And here is a shout out to SRSLY. We use the Pico Plus kit to actually sequence for our cut-and-run experiments, where cut-and-run is a epigenomics tool that tells us where HSF1 is located on the genome. And when we do so, we actually see in this meta plot shown here, which is centered around heat shock elements plus or minus 1 kb, that compared to unstressed condition, that there is a dramatic increase in HSF1 occupancy at heat shock elements across the epigenome upon heat shock. However, upon sodium arsenide stress, which is those type 2 stresses that I showed you where the transcription hubs are stalled, the occupancy is decreased. We can now correlate this with nascent RNA expression via BRUSeq, which is a metabolic RNA labeling method, followed by sequencing, where again, I'm showing you here a meta plot of genes which are induced by HSF1 across this transcription start site, plus or minus 2 kb. You can actually see that those genes are actually, our target genes are induced upon heat shock, but the extent of induction is much lower upon these sodium arsenide stress, which again is a representative of those type 2 stresses. So in a nutshell, we asked a question, do HSF1s drive enhanced gene expression during stress via condensate formation? And the answer is not simple. It really depends upon the context. So in stresses like type 1 stresses of heat shock, chaperone, or proteasome inhibition, you get this beautiful enhancer hub formation, which is potentially correlated with cell survival. However, on the other hand, in response to these type 2 stresses, you actually get sequestration of HSF1 within transcriptionally dysfunctional complexes, which is highly correlated with cell death. So with that, I will stop. And I would like to thank all the wonderful people who have done this work. Folks highlighted in green are the ones whom we collaborated with for the prostate cancer and the castration work. The ones highlighted in red are the ones we collaborated with on the HSF1 work. Specific shout out to the Matthias Trutman lab, who worked on some C. elegans models, which we did not have time to show. Mats Lungman and the BRUSeq core, who are involved in the nascent RNA sequencing, and Srinivas Ramachandran and Giovanna, who are involved in the cut and run sequencing. I would like to thank our past and current funding agencies, as well as our lab mascot, Strauss. Strauss rocks on in spite of stress. With that, I'll be happy to take any questions.